We'll just have to wait for a second for it. Okay, we are live. Okay, so I would like to welcome all of you on this a pleasant Wednesday afternoon as I look out of my window at TIFR Mumbai, and I really give a warm welcome to Professor Sudha Rajamani, who is joining us from IISER Pune, from her office for this particular colloquium. And I also want to welcome all of my colleagues and students for this very interesting session. Sometimes we rarely get biology seminars, but this is actually connecting everybody's work across something on origin of life. So before I formally introduce Professor Sudha Rajamani, I would like to mention to her and other people who are listening to this colloquium for the first time. The Wednesday colloquium is as old as TIFR itself, and it was started by our founding director, Professor Homi Bhabha. The idea was to get all the scientists working under one roof, physicists, chemists, and biologists, to get together once a week at 4 o'clock on the Wednesday and listen to lively expositions of fantastic talks. It's my honor to be part of this colloquium series and conduct this. One of our old colleagues who conducted this, older colleagues, Professor P.K. Madhu has joined us from TIFR Hyderabad. Madhu, welcome. He had been conducting it for many years since I joined. So Professor Sudha Rajamani actually comes to us from IISER Pune. And her work, actually, her lab's work really is trying to enunciate the chemistry part of origins of life. Okay, that's her lab's title is Chemical Origins of Life. Professor Rajamani actually got her PhD in biochemistry from NII, New Delhi. And in her PhD, she worked on sickle cell anemia and the hemoglobin associated, the biochemical properties of hemoglobin associated with sickle cell anemia. Once she finished her PhD at NII, she went to Tony Fink's lab in UCSC, which is University of California, Santa Cruz. And that is where she actually started looking at alpha-synuclein's role in Parkinson's disease. However, there were things going on during that time when she was with Tony Fink, and she moved on from that lab to, I guess, Professor Dreamer's lab. Is that correct, Sudha?
Yeah,
David Dreamer's lab in biosciences, biosciences in UCSC. And that is where she claims that her love for astrobiology started. And after her work there, she went for a short postdoctoral stint at Harvard, and she joined IISER Pune in 2012 as an assistant professor. She has been there since, and her work is clearly enunciated. Last year, she had a series of papers which came out, and this year also, which is showing that you really need to understand as simple as a membrane, lipid, and you can start putting together compartments that were so critical in the origin of life. Of course, the questions of origin of life is not just for our planetary system, but is also for the entire universe. And if we can detect such life somewhere else, there is a famous paradox, the Fermi paradox, which also relates to this idea that can we detect extraterrestrial life. And if it were already there, they might have detected us. So this kind of a context, which is a very broad one, I invite Sudha to give her colloquium. Sudha?
Thank you so much, JD. Thank you. Thanks to you and Ullas for making this possible. It is a terrible shame that I'm not able to do this in person, but I'm really glad to be nevertheless connecting with all of you via Zoom. And like I already alluded to you, I am totally going to encash the ticket of that exquisite sounding lunch in person at some point, which goes with giving a TIFR colloquium. I think I'll start off with sharing my screen, if that's okay.
Yes.
Yeah. Can you see this PowerPoint presentation?
Yes, Sudha, thank you. Yeah.
So today I'm going to try and give you folks a flavor of what it is that we do in lab and what is sort of the context of the work that allows me and my colleagues in the field to be able to undertake ask specific questions in this area of research. And eventually, the hope is that I will be able to present to you some of the pieces of the puzzle that is finally coming into view. Do we know completely about everything that happened? The answer is no. But can science explain the origin of life? The answer is yes. And how this story might have panned out or would have panned out on the early Earth is what I would like to give you a flavor of. These are essentially the grad students who have either gone through my lab. Chaitanya and Nirja were the ones who helped me get the lab up and running. And then these are the current grad students and one postdoc who are vital to helping me pose these questions in the lab and try and systematically figure out the answers. And I really have to acknowledge that a whole bunch of undergrads also have been through our lab, and it's been a fun ride thus far. And yeah, let's start. So one of the important things that I wish to highlight is this absolutely undeniable continuum that exists between physics, chemistry, and biology as is illustrated on the slide. So if you basically look at the bottom, you have a bunch of biomolecules, and these are these more complex protein and nucleic acid-based molecules, which are cytochrome C and hammerhead ribozyme respectively, which are made up of simpler monomeric entities known as either amino acids or nucleotides. But eventually, essentially, if you look at them, what are they? They are a bunch of atoms strung together into a molecule, and these molecules come together to form much more complex molecules. And somehow this transition from this simpler chemical space to a more complex chemical space is thought to have laid the foundation for the transition from non-life to life. And you now zoom into these molecules, and what do you see? You actually see even more simpler molecules, and some of these molecules are very readily formed in our interstellar medium. So it is not unique to Earth's chemistry at all. Molecules like amino acids and sugars have been found on riding on meteorites that essentially have landed on the Earth, and people have been able to systematically look at them. People have also extracted entities which...
Sudha, sorry, this is Jyotishman. I'm just interrupting you. There is a little bit of concern about the audio. Could you just be closer to the mic? I don't know if that would help.
I have no clue, but let me... Is it okay now? Can you hear me?
Yes.
Is it better?
Yeah, that's better.
Okay, sorry about that.
Thank you, Sudha.
So the idea is that these molecules are very readily made in the interstellar medium, and more recently, observations from the Hubble Space Telescope this happened between 2016 and 2018. These observations actually have gone on to increase the upper threshold of the size of the molecules that can be found in the interstellar medium, and that is this particular molecule shown here. These are called buckyballs. They were named after a famous American architect called Buckminster Fullerene. And it was mind-blowing for the scientists at the Goddard Space Institute to actually systematically be able to see them, because prior to that, even being able to find a molecule which is made up of C3 or C4 carbon atoms is non-trivial, because you're looking at a zoo of peaks in the spectrum. And to systematically assign these peaks is one challenge. And the other challenge is to actually discern for sure that these are indeed peaks that are coming from the interstellar medium and are not being sort of diluted by spectra that is otherwise being sort of recorded by the atmosphere. So in this sense, the Hubble Space Telescope is outside of the Earth's atmosphere, so it partially sort of provides a respite. And this particular thing basically sort of blew all of us when we could see that for the first time it was shown that you can see molecules as complex as C60 carbon atoms readily sort of forming in the interstellar medium. Zoom into them, what are they? Fundamentally a bunch of atoms. So this continuum from physics to chemistry to biology is so obvious that one of the immediate things that stands out to you is, if you do wish to understand life, the big question is to ask, how did chemistry transition to biology? And to sort of set the stage for this, how do we think these biomolecules could have been found on Earth billions of years ago? And we're talking about billions of years, or on the order of 3 to 4 billion years ago, right? So it's thought that these molecules could have come onto the Earth could have been present on the Earth because of two processes. One is exogenous delivery, where they can ride on extraterrestrial objects like meteorites and comets. And even today, these things bombard our planet and carry with them a lot of these interesting chemical entities. So one of the ways this would have happened is via exogenous delivery. And more specifically, there's thought to have been this late heavy bombardment period around 3.8 billion years ago. It's being, I'm told it's a little debated in the planetary sciences field. Nonetheless, there is sort of a strong sort of notion that this was indeed a phase. Whether it was a very concentrated event or an event that was spread out is still debated. But suffice to say, a lot of these molecules could write on these extraterrestrial objects and be delivered onto our planet. Actually, more readily, you can also invoke terrestrial syntheses of many of these molecules that I'm showing you in this panel here below. They can form, for example, decanoic acid and caprylic acid are fundamental monomeric units that make up your lipid membrane, right? These are fatty acids. These molecules can form via a process called Fischer-Tropsch type syntheses, which can readily occur under high temperature and high pressure conditions on the Earth's atmosphere. And you require the presence of carbon monoxide and water for these molecules to form. So in these two manners, it's thought that molecules relevant to biochemistry, as we understand extant biochemistry, might have been readily available on the early Earth. So then,
just one quick question about this.
Yeah,
yeah, these are good hypotheses. There's no question about it. But the fact that the timescales of such sort of reactions and so complexities to develop, do seriously people believe that you can actually have long chain hydrocarbons generated by one such event, or it requires maybe the initial event that seeds it, and then continuous chemistry on the surface of Earth would lead to generation of future larger chains?
So when you say seeding, are you referring to exogenous delivery based seeding?
Exogenous delivery, delivering maybe the first fragment of carbon 1, carbon 2 fragments, and then expanding on the planet.
So the real answer is, we systematically haven't had this debate in the field, but like, just to kind of sort of give you a tangential response, labs in the US that have actually systematically looked at FTT type syntheses can actually start from the fundamental ingredients and readily form up to close to C12 sort of fatty acid, hydrocarbon chain lengths. So this is something that people have shown.
In one event, in one such event?
No, so they do some sort of cyclic sort of experiments. I do not know the precise details about this, but I'm happy to find out and let you know.
Yeah, no, it would be nice to know the time scales of such synthesis, because
true.
It could be hours or days or years. That's the question.
No. Oh no. So this is more like on the scale of days, not hours, not years.
Fine.
On the scale of days. And given that we've had enough time, billions of years, right, to have events play out, I think this would have not been the rate limiting step. But even if the terrestrial syntheses, like you allude to, and interestingly, I can immediately think of some experiments how we can show whether these reactions can be sort of catalyzed in the presence of fundamental seed dimeric units. Even if this were tricky, exogenous delivery seems to be very readily plausible. One of the examples is Murchison Meteorite, which fell in Victoria in this village called Murchison back in the 60s. And my mentor in the field was one of the first people to look at the core of this meteorite to study the organic composition of the meteorite. And they could demonstrate actually C8 to C12 fatty acids in the core of the meteorite, which could readily assemble into higher ordered structures, just like our fatty acids, chemically synthesized fatty acids can do in the lab.
Okay.
So this was later on systematically shown by other high-end mass spectrometric methods that it indeed is this. So yes, maybe it was a bit of both, but maybe a lot of exogenous delivery. It's unclear. Given that you could have these processes populate the relevant molecules, then the question to ask is, how do we think about the actual transition? And one way to think about this question is indirectly sort of, is the answer to what this slide depicts. I'm showing you a bunch of pictures here, all the way from a bacterium to Nelson Mandela with T-Rex thrown in there, right? So what is sort of the least common denominator that unites all of these pictures? Cells. If I gave an outreach talk, I would wait for the students to answer, but right now this is not one such talk, so I'm going to reveal it. Cells. So just even though cells were first observed soon after the first microscopes were discovered back in the 17th century, it wasn't appreciated till about the 19th century that they are the fundamental unit of most life. And on this slide, I'm showing you representative images of prokaryotic and eukaryotic cells. And I think the take-home message for me from this is that even these cells are extremely complex. And to think about how they might have readily emerged from a soup of interacting molecules called the prebiotic soup is completely non-trivial. So then how do we, and the other amazing thing about this, and even these molecules did emerge, right? We have a timeline. The eukaryotic cells are thought to have emerged about roughly 2 billion years ago. Prokaryotic cells, some of the oldest signatures are about 3.85 billion years old. They've been found in stromatolites in Australia, on the western coast of Australia. Since their emergence, what have they been capable of doing? They've been able to do this. They've been able to populate every single niche of life. We are talking about single cell containing microorganisms, entities, but they are so cool that they have been able to adapt and populate every extreme environment that you can throw at them. Permafrost, they're found in extremely cold environments. Halophiles, salt-loving organisms, UV-resistant organisms, and extremophiles or thermophiles, which can thrive even at temperatures close to 90 to 100 degrees. I think one of the organisms that's been shown to tolerate the highest temperature now, the record is around 120 degrees centigrade. So then it becomes much more easy if you pose the question to try and understand how did the most fundamental unit of all life come about from your prebiotic soup. Here is a very minimalistic cartoon depiction of the tree of life, which was inspired by Darwin's theory of evolution, right? Which essentially meant that all of the complex entities that populate these three branches way back in time probably emerged from this single entity, which is called the last universal common ancestor, or LUCA, or now it's called the last common ancestor also. And what is interesting is, even though Frederick Wohler's demonstration of synthesizing urea in a test tube for the first time showed that, for the first time shook the fundamental core of the philosophy of vitalism, which prior to that attributed life to being this magical force that only the living forms harbored. The fact that Frederick Wohler could actually synthesize urea, which was otherwise extracted from human urine, readily in a test tube, shook this particular vitalism theory to its core. But nonetheless, nobody systematically still started thinking about origins of life soon around that. What was a bigger sort of focus was the evolution, the theory of evolution as proposed by Darwin in his Origin of Species book. But the one thing that I want to kind of put out there is, when Darwin was talking about all of this, right? He still was talking about thinking about the origin of life question, even though he didn't articulate it, because I think he had already pissed the Christian folks enough, and he didn't want to throw another theory in there that would have pissed them off even more, right? But he did discuss this in a letter which he wrote in 1871, and his language is so excitable, right? You can sort of see he knew that this question is super deep, and this is me reading out what he said. But if, and oh, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity present, that a protein compound was chemically formed, ready to undergo still more complex changes. he almost sort of articulated the hypothesis of the chemical origins of life back in 1871 without realizing that this was a very valid hypothesis. Happy to say that since then, a lot of work has gone on in the mostly in the early 20th century, and now this tree of life no longer is thought to look the way it looks now. This is a more sort of a realistic vision of the tree of life, and you can see that the domains exist as they do today, but there's a lot of crosstalk happening between all of these domains. And it is thought that way back down, very early on in the history of life, you did not have probably this one last universal common ancestor, but you had a common ancestral community of primitive cells that sort of behave a lot like how your modern cells behave, where they constantly talk to each other and do a lot of things in conjunction with each other, so on and so forth. Sorry about that. Yeah. So what are these fundamental units thought to be like, right? These are called protocells or primitive cells. This is fundamentally a lipid membrane which encapsulates some form of genetic material or some minimal metabolic network that allows this cell to sustain itself. So this brings me to a very important question, which is crucial to sort of put out there. When we think about the chemical origins of life, we need to work with a working definition of life. And if you actually go and look for the definition of life, you're just going to get a crazy number of responses when you sort of Google for it. So what happened was the NASA Astrobiology Institute constituted this committee which came up with a working definition of life. If we look at all of life that surrounds us, we can sort of list about 6 or 7 characteristics that are crucial for something to be for something to be referred to as life. But even within those, there are even more probably certain minimal fundamental concepts that are ultimately crucial, right? So the definition that we work with is that it is a self-sustained chemical system capable of Darwinian evolution. Sort of that is the working definition of life that we sort of work with. And this particular image is a artist's representation of a protocell, which gives you a sense of what we think the most primitive forms of cells might have looked like. And what can we do? The best we can do is to draw up this story, which is consistent with all the evidence that we have that come from experiments that we do in the lab in areas of chemistry, geology, so on and so forth. What we understand about what the early Earth looked like, and very importantly, what biology, extant biology, reveals about some of the oldest forms of life on that tree of life, right? So happy to say that after a century of lot of efforts, the story is finally coming into view in bits and pieces. So this field is called astrobiology. It's basically a field that tries to understand the origin, the distribution, and the evolution of life, not just on Earth, but in the universe. Back in the days, there were 2 fields, astrobiology and exobiology. Astrobiology sort of restricted itself with understanding how all of this panned out on our planet, and exobiology was meant for what happened off the planet. But since then, in the last decade, there's been a change, and this definition now encompasses life as we understand on our planet, but as it can be found anywhere. It is a highly interdisciplinary area of science. And as this particular sort of image shows you, it feeds off of inputs from a lot of different areas of science. And we in the Cool Lab sort of work at this interface, chemistry, biology, and geology. And within this by using tools in these areas and by collaborating with, theorist friends who do modeling, and we also do some, we use tools in biochemistry, mol bio, biophysics, chemistry, to fundamentally try and understand how can you go from the prebiotic soup of molecules to a protocell. That is sort of the big focus in the lab. So how does this timeline look? This is sort of a very crassish timeline of how we think about the early history of life on Earth. Not all of the nuanced processes have been timed or sort of situated very clearly on this timeline, but suffice to say, you get a good sense about what might have been. So 5 billion years ago, formation of Earth happened. Then subsequently, around 3.8 to 4 billion years ago, Earth's crust formed and the oceans sort of would have populated the planet, allowing for very interesting prebiotic chemistry to be plausible because of all that amazing chemical molecules that were delivered onto the early Earth. Now, from this space of prebiotic chemistry or the prebiotic soup, how did the transition from here to biology happen? And how do we think about it? Extant life or modern biochemistry has three fundamentally important biomolecules, right? You have this genetic molecule, which is the nucleic acid, DNA, which encodes all of life's information. You have proteins, which are the catalytic molecules, the workhorses of our cells. And then you have lipids, which are essentially the membrane organelles, or the compartment, the entities that provide this compartment that separates the inside from the outside, which is super crucial for allowing related molecules to interact with each other. But very early on in life, it is thought that you didn't have to have all of these molecules come about to be able to sustain the most primitive form of life. And why do I say this? This is because of this amazing molecule called RNA, which is a cousin of DNA, which is not only capable of encoding information, but it's also capable of catalyzing reactions in the form of ribozymes. And if then one can simplify this problem of trying to understand how do you go from the prebiotic space to an RNA world and understand these processes with a clear understanding that there was a contribution of lipids that would have come into play that eventually allowed for the emergence of protocells, we can then think about this transition in a much more systematic and in a stepwise fashion. And what do I mean by this pre-RNA world here? In the last couple of decades, it's become very obvious that going from this prebiotic soup to an RNA world is completely non-trivial. And you would have had a time in the early history of life where this RNA world was predated by more simpler molecules, which would have much more readily emerged from the prebiotic soup and eventually evolved or would have been outpaced and the RNA world. I'll touch upon some of these when I'm talking about some of the work that we do in our lab. But when did all of this?
Question? Can I ask a question?
Yeah.
Is there some sense of where multiple reactions inside a compartment started? Is there some sense of that? Because
you mean the niche?
You're giving the tools. You're talking about all the tools that are possible to do a reaction. But when did you think, or where was this in this timeline? Complex reactions talking to each other in a compartment really happen?
See, here's the very interesting thing. You would have had potentially interactions between the membrane component and the other molecules, because they were also components of the prebiotic soup, right? They were very readily found there. But what is interesting is, given that all of life's chemistry or life's biochemistry is in the context of water, lipids very sort of immediately form these higher ordered structures. Right. And when they self-assemble into these higher order structures, they facilitate a lot of interesting possibilities in the prebiotic soup itself. Okay. So there might have been a lot of crosstalk that would have happened right at this stage, which then allowed for them to sort of encapsulate some of the growing oligomers that can form in the subsequent stages. I'll touch upon it with when I talk about the model that exists for a terrestrial origin of life in a couple of slides down. But it's a very important question. So when did all of this come about in terms of how the theory was put forth and what the hypothesis was out there? It's super cool that the theory for the origin of life, chemical theory, for the chemical origin of life, was put forth by two scientists who either came from a background of atheism or were atheists. Yeah. So Operan, who belonged to USSR and who actually was a avowed communist and was also conferred the Order of Lenin, basically was one of the first people to sort of clearly describe this hypothesis of how can you go from chemistry to biology, that if you had this prebiotic soup with all the right ingredients and the right energy sources were available for these molecules to interact and form more complex chemicals, it is that sort of space in which this kind of transition could have happened. Almost around the same time, maybe give or take 3-4 years apart, Haldane, who has a very interesting India connection, separately, who he did a lot of interesting work in the evolution, in the theory of evolution, and he was also thinking about this question, and he also, for the first time, proposed this concept of primordial soup, where fundamentally, given the fundamental ingredients that is important for the molecules that are relevant to life's biochemistry to emerge. If they were present in this primordial soup, and you had energy sources like lightning, which was very readily plausible on the early Earth, super hot, and a lot of interesting volcanism happening, and a lot of high energy events happening. You had ultraviolet radiation from the sun. Given these conditions, you can have these molecules, inorganic molecules, interact with each other and spontaneously form molecules that are more relevant to extant biochemistry. A theory is a theory till it's proved otherwise, right? So this particular theory did exist from the mid-1920s for a while. It wasn't demonstrated till Miller-Urey experiment actually happened in 1953. Miller walked up to Harold after a talk and said, hey, I'm fascinated with this theory and I would like to sort of demonstrate, I'd like to check if this hypothesis is indeed correct. And Harold was like, please, well, you're welcome. I'll be happy to support you. And Harold actually was giving a talk on chemical origins of life. And the two of them banded together. And this is the very famous Urey-Miller setup where Stanley went on to show that if you started from a bunch of ingredients shown on the left here, methane, water, ammonia, and dihydrogen, and if you could simulate the lightning on early atmosphere, right, you could actually have these molecules interact with each other to form readily some of the organic molecules that are the fundamental components that you and I are made of. And this was the experiment that for the first time demonstrated that the hypothesis that was put forth by Oparin and Haldane was indeed plausible. This demonstration of this experiment and subsequent cool work that came out of Stanley Miller and Urey's collaboration set the stage for the field of prebiotic chemistry to come about, which is the area in which we work right now. So very broadly, what are the steps leading to the emergence of a primitive cellular entity? A lot of interesting prebiotic chemistry allowed for the formation of building blocks. And what is important to remember is the building blocks or the monomers can form very readily. What is super difficult is to envisage scenarios and think of processes where they went from these building blocks to polymers, which is what life is made of, right? Your protein is a polymer of amino acids. Your nucleic acids are polymers of nucleotides. Your lipids are higher ordered self-assembled components, which are made up of multiple units of lipids. Your carbohydrates are polymers of simple sugar monomers. So it's extremely important to think about how you can go from this monomeric space to a polymeric space. And when you think about this transition, there is this chicken and egg problem that one has to contend with. And what do I mean by this? If you look at modern cells, proteins are the workhorses of all cells, which allow us to do a whole bunch of things, right? Metabolize reactions and essentially allow us to live and survive. These proteins, they are made from the information that is encoded in the nucleic acid. But in order to make the nucleic acid, you need the proteins. So which came first, information or catalysis? That is a very big question in the field, right? So in order to sort of, the RNA world hypothesis provides a wonderful respite from this problem because you can actually invoke non-enzymatic mechanisms where you can try and understand how completely in the absence of these elaborate and awesome machinery called proteins, you can still try and understand how can you go from this monomeric space to a polymeric space. And once you have the RNA world polymers come about, the other next important question is to think about is how does one envisage that the information encoded in these polymers were replicated in these very harsh conditions of the early Earth, because you do not have enzymes, you do not have these machineries that allow for extremely high fidelity reactions to happen in your cells and my cells. So then that is something to think about. And it's very important to understand this mainly because if in this RNA world space, you did come about with catalytic molecules, which you can, because you can do these experiments in the lab and show that you can evolve ribozymes from a pool of starting mers. Then the question is to try and understand how might have these ribozymes replicated the information encoded in them, because you would otherwise lose out on their function if the information gets garbled. So one of the other next important step to think about is to understand non-enzymatic replication and how might have it been facilitated. And all of these steps we now know are very closely interrelated to the presence of a very important co-solute in the prebiotic soup, which is that of membranes. It seems to play a very important role in all of these processes and eventually has a bearing on the emergence of the earliest cells or the protocells. So the environments that would have supported these life-producing chemical reactions, depending on whom you talk to in the field, it's either these hydrothermal vent systems, these volcanic systems which are present on the ocean floor, or these volcanic systems which are present on the terrestrial atmosphere. We in the lab simulate these terrestrial geothermal pools in the lab, volcanic geothermal pools, to try and understand the questions that we are interested in. Those who, excuse me, those who work in this realm also have to worry about one other question, one other parameter, important parameter, which is that of pressure. In our case, we don't have to sort of worry about it much. So these are some niches where we think some of these things might have happened. So the model that we work with is as follows for the terrestrial origins of life, right? If you had these volcanic geothermal pools or these tidal pools, and if you had the monomers that are important to eventually form these protocells, as I already alluded to, your lipid molecules, one thing to please remember is that our modern cells are made up of very complex lipid molecules. There's a whole variety of them, and they are synthesized using heavy-duty machinery that is encoded by our genes. So one has to sort of, in order to make it much more readily understandable or to make it much more readily plausible, have to think about or invoke simpler amphiphilic entities. And here are what I'm showing. These are fatty acids. These are very much present in our phospholipids. These are the single chains that come together along with the head group to form the more complex phospholipids and the other kinds of more complex lipids in our cells. But just these individual monomers by themselves can very readily assemble into higher order structures. And if they were present, they have these water-hating groups. So as soon as they see water, they will ball up into these higher ordered structures called micelles or vesicles. Vesicles are basically formed after this biliary stage happens, and they folds back on itself to form the vesicles. So during one of the important things is to think about the energy source that I was talking to you about, right? And one of the very readily available energy source that would have been present on early Earth is day-night cycles. You have hot days, cold nights, and this would have facilitated wet-dry cycles, just as it facilitates even on modern Earth. It would have done this much more readily on the early Earth because of the higher temperatures. So if you had these monomers of the amphiphiles, and these red guys are supposed to depict the monomers of the informational molecule, if you had them in the same space, a cycle of dehydration makes these higher ordered lipid structures to collapse back on itself to form these multilamellar sandwiches, which now entraps these monomers within the interlayers. And here is a real freeze fracture image of these multilamellar sandwiches. And once now you take these monomeric entities, you constrain them into a two-dimensional space, they can interact with each other much more readily than, say, in a three-dimensional space. So one thing to importantly remember is the following. Going from monomers to polymers is a non-trivial problem because you have to go from a thermodynamically stable state to a thermodynamically unstable state. So you need to, in modern, extant biochemistry, all of this is facilitated because of the way our biochemistry is structured, and a lot of enzymes sort of take care of these problems. But on the early Earth, what might have the mechanisms been that would have allowed for this? And this is one of those very readily plausible mechanisms where you now are constraining the monomers from a three-dimensional space to a two-dimensional space, which then allows for ready interaction of the molecules in this space. There is still some diffusibility that is possible, especially because you have lipids here, and they can form these oligomers. And these now, on a subsequent rehydration phase, get entrapped within the blebbing out membranes, like it's shown here. This inset, which shows these blebbed out membranes, is actually a decanoic acid decanol 1:1 sort of composition-based membrane. And these processes are very readily demonstrable even in the lab. And it's thought that under these multiple wet-dry cycles, you could have facilitated some form of selection, which would have selected for the more stable protocells. And these kinds of cycles would have essentially, eventually, allowed for the complexification of the primitive cells that formed in these kinds of atmospheres. This is just a artistic illustration of what I just talked to you about. Dehydration.
I have a quick question here.
Yeah,
So,
yeah.
when you think of early Earth and water, do you consider this water to be like neutral pH with no ions in it? Or is
Very,
this
very, very important question, right? So I think the other way to articulate your question is to say, under what pH did all of these reactions, at what pH were they facilitated, right? Very important to remember that all of this geology here will essentially constrain the physicochemical parameters of the solution that's present here. Depending upon the rock chemistry, you're going to have either an acidic environment or a slightly neutral environment or a basic environment. Just like on modern Earth, when you go to these hot springs, you see hot springs which are just a few feet apart that can be tremendously varying in their pHs. One would be, say, at 2.3, and just a couple of feet away from that, you have another system that can be at a neutral pH. So just like that, you could have these volcanic geothermal pools where, depending upon the rock chemistry, the pH of that limited space would have been subtly different from the others. This kind of polymerization reaction that I'm specifically talking about, for example, if you want to join a bunch of nucleotides into a nucleic acid, you will have to invoke acidic conditions to be able to facilitate the stitching of the internucleotide linkages. It doesn't happen readily at neutral or alkaline pH, and I will show some data that shows that. So yes, it's very important to think about the physicochemical parameters of the space and think about the pH of the solution in which all of this is occurring. But let's assume all of this was indeed possible.
Okay.
Then you can, sorry, yeah, tell me.
No, please continue. There are two other questions. I'll come to that.
Okay. So basically, it's exactly what I talked to you about. You can have this stepwise evolution of these polymers that could potentially form in these scenarios, where if you say had these multiple mutated molecular systems with a focus on A and B, and system B, for example, is a random pool of a random bunch of protocells that encapsulated this B molecular system, which doesn't really confer a selective advantage onto the system. This particular protocell will just disrupt and vanish. But certain other molecular systems might have provided a selective advantage to the membrane that encapsulated them, and it is thought that these kinds of cycles would have essentially finally led to the complexification of the protocells that can be formed in these environments. So
let me just take these questions.
Yeah.
So there is one question by Arya Nachiketa.
Yeah.
The question is, can study of virus unravel the transition from non-living to living matter?
One of my favorite questions. I talked about a definition of life, right? What did I say when I sort of constrained that definition? I said life is a self-sustained chemical system capable of Darwinian evolution. Just by this definition, virus just lies a little outside of it. It is not a self-sustained chemical system, right? It needs another living system to be able to sustain itself. So depending, a majority of the people in the origins of life field do not classify viruses living, but there is a small subset of people who very strongly feel that we need to describe a fourth domain in the tree of life, which is made of viruses. So yeah, to me, the answer is that it doesn't even fit within the constraint of the definition of life. So to be able to think about it as a transition, very early, an entity which probably emerged very early on during the transition, possible, but it in itself is not going to fit the definition because it's not a self-sustained chemical system. So I hope that has answered the question. You said there's another question.
Yeah, so Alak Ray has a question. Alak, you could go and ask the question. You can unmute yourself.
You have been talking in terms of this monomer to polymer formation about dihydrogen. Now, suppose I had a small fraction which is actually variable from planet to planet of the other isotope of hydrogen, namely deuterium. So would that actually affect how this formation of polymers process would happen? The question is relevant because, for example, in Venus, the deuterium to hydrogen ratio is much larger than than Earth's ocean, and so could it have been constraining factor for Venus, for example?
Alak, right? So, we do not have an answer to this question. Theoretically, it seems like it might potentially impinge to some extent. To what extent is unclear. The people who have tried to revisit the Urey-Miller experiment, and there is a whole set of colleagues in my field who are trying to recapitulate that and try to play around with variations. Some of them are actually precisely looking at this. We still do not know what the outcome of these experiments are. I met them about a year and a half ago in Vienna. And this was one of the questions they had, because they were teaming up with a bunch of planetary scientists. And this was one of the things that they were keen, that if you change the isotope nature of the isotope that you're working with, will that have implications? So I really do not know the answer, but it feels like there could be some effect. How dramatic would it be is anybody's guess.
Okay, thank you.
Should I go ahead? I can't hear you. But
so there's one more question, which is when and how did virus originate? So you could take this now or later.
Yeah, I really don't know, but we'll get to it later. Maybe this is more of a debate than, yeah. Okay, so to move on, I'd like to sort of highlight that since the field of prebiotic chemistry came into existence in in 1953 specifically, a lot of work has gone into understanding whether you can have a recipe for life emerge from the same set of starting ingredients. Even though multiple colleagues in the field have been able to demonstrate multiple precursors different precursors leading to the different classes of the fundamental monomers, a very cool piece of work from Sutherland's group has actually shown that this is based on a cyanosulfidic metabolism-based set of experiments, where you can have the same set of starting ingredients and you can form precursors that eventually lead to the formation of amino acids, nucleosides, and lipids. So we have come far enough to the point where we understand to a much, we can reliably say that prebiotic chemistry did lead to the formation of monomers. One of the next important questions is to try and understand how you can go from the space to the polymeric space, because that seems to be fundamental to understanding how this transition might have happened, or how these things emerged in the first place, started interacting amongst each other to sort of possibly depict the common ancestral community of cells that I showed in the second tree, which is what we think more the tree of, which is what we think is a more realistic depiction of the tree of life. And then how did they then evolve to the space where 3.8 billion years ago, you had prokaryotes emerge on the early Earth. So I won't get into this, but emergence of prokaryotes had fundamental implications for the Earth's atmosphere, in the sense that they helped oxygenate the Earth's atmosphere. That particular event would have certain other consequences. I'll not touch upon that, but this was fundamentally crucial for allowing the kind of complexity that we now see on early Earth to have been generated subsequently. Yeah. And these events are thought to have happened about 3.5 billion years ago. And here you're looking at stromatolites from these pillow-like formations. These are found in Perth in the west coast of Australia. And these are essentially formed as a result of interaction between microbiology and geology. And these are some of the oldest signatures of life that are found on the Earth today. And around 2 billion years ago, the more complex nucleated cells, which you and I are made of, are thought to have emerged, which eventually led to all of this greater complexity that we see in the tree of life exists today, happening after that, after the 2 billion years ago event roughly, right? So all of us in the field hope that this is going to be the reality someday, right? You're going to do all of your fun experiments, and someday this little thing will come up and say, "Are you my mommy? Are we there?" No, we aren't there. In our lab in particular, we're interested in understanding multiple aspects of this problem. One is the origins of both the both the informational molecules and the protocells, the most primitive form of cells. We are also interested in understanding within the informational molecule space how non-enzymatic replication or transfer of information would have been facilitated, because it's very important to think about the fidelity of these processes, the kinetics of these processes, because they could have been affected by other molecules that are present in the protocell, in the prebiotic soup. And then we are also very interested in understanding how the most primitive form of catalyst might have emerged, particularly when we think about a lot of the enzymes that we see in biology today. You have at the core this metal mediated center, which is either directly going to be playing an important role in the catalysis or has a secondary but super crucial role which eventually facilitates the catalysis to happen. So this is another sort of area that we're very interested in. And we are also very cognizant of the fact that a lot of the work that we do in the lab and in the field has important implications for trying to understand whether lifelike processes might exist outside of our planet. And we do this in conjunction with a whole bunch of other colleagues in the field, both within India and outside India, and many of them who don't even call themselves astrobiologists. I will use two examples to talk about how we think about the problem of the emergence of protocells. So you need to think about two things. One is about the informational polymer itself. Think about the compartment or the sort of the sac-like ball in which all of these molecules are interacting with each other. So in this context, we have been very interested in understanding how the molecules of an RNA world came about. And in modern biology, this is a very complex process which involves a whole bunch of molecules which are super complex, right? But the RNA world hypothesis basically posits that you need to sort of invoke completely chemistry-driven mechanisms, otherwise you're going to run into the chicken and egg problem. So this constrains the possibility that your polymerization had to be non-enzymatic very early on. So how do we think about this? We think about this essentially as facilitating condensation reactions that would have allowed for the conversion of the monomers into polymers. Here is two nucleotides, purine nucleotides, which are condensing with each other, losing a water molecule to form this phosphodiester bond and form a dimer entity here. What has been happening in the field? People have tried to understand this problem, but they do something very important. Just now, this particular thing that I showed you, right? This purine nucleotides right here, these are what we find in our cells. These guys are very readily stitchable into a polymer when you have enzymes. If you don't have enzymes present, it's extremely difficult to work with them. So what do people in the field do? They kind of activate the monomer, They put a chemical entity on the monomer that makes it much more reactive, that makes it much more excited, so to speak. So these are imidazolide containing activated nucleotides, and people have looked at them in great detail and tried to understand various methods of polymerizing these. They've used clay catalyzed polymerization. And why is clay interesting? Clay is a product of volcanic degassing. So it is actually even found on other planets of our solar system. For example, Montmorillonite is this rock star clay which very readily facilitates polymerization, and it's been found on Mars. So it has interesting implications for trying to see if you can find these kinds of oligomeric signatures if you kind of bring back some of those samples, right? Here is showing you this interlayer in the clay sort of in a unit where you can have your nucleotides line up. Again, a two-dimensional constraining of starting reactants is happening, which facilitates crowding and which facilitates uphill polymerization reactions to occur. And depending upon the clay, you can have a good clay, you can have a poor clay, you can have a excellent polymerizing clay. People have shown these even in eutectic phases, which are formed when you have salts and water. You reduce the freezing, point of the water, and at the point where it freezes, you have, other than these crystal spaces, you actually have these brine channels where there's a lot of salt concentrated. And if you have monomers in these spaces, they very readily oligomerize. It is not enough that you just take water and reduce its temperature and this will happen. You need these brine channels, and salt also is a fantastic condensing agent. So even though, and hence their presence is super crucial for facilitating these reactions. But now coming to non-activated nucleotides, which are more realistic, because these activated molecules that I talked to, I showed you only one molecule. There are a few out there that the people in the field think about and talk about. The problem is to think about the chemistries that might have resulted in their formation. It's much more easily feasible to think about how non-activated nucleotides, like the ones in our cells, might have come about. Polymerizing them, very difficult, but if you figure a way out to do it, it's much more realistic, right? And this is where those fluctuating environments are fantastic. Molecules are not inherently reactive, but the environments in which they were present facilitate these wet-dry cycles, which allow for condensation to happen during the dehydration stages. So it could have promoted uphill polymerization reactions. The geology drives the chemistry of that niche, which, as Vamsi had posed that question, plays a very important role in deciding the kind of reactions that can be facilitated there. And this is, again, just a depiction of what these niches might have looked like. A lot of exogenous delivery happening, but a lot of terrestrial chemistry also feasible, which is where these reactions might have been driven. High temperatures, varying pH, the number of dehydration, rehydration cycles that the reaction sees, all of this could have a bearing on the reaction. And these physicochemical parameters, which can naturally be facilitated in these scenarios, lead to this environmentally driven kinetic traps, which then allow for uphill reactions like polymerization to happen. So we in particular have looked at lipid-assisted synthesis of RNA oligomers. This was demonstrated in 2007 as being possible, that when you have lipids and monomers, you can do multiple wet-dry cycles, form these, sorry, I ran through it, but you can form with increasing number of cycles. You see how the color of the streak increases. You can actually form longer oligomers with time. And this is because these multilamellar sandwiches can actually entrap these monomers, which can then undergo condensation reactions to form oligomerization. So you can have an organizing effect coming from this. And the good thing is, on a subsequent rehydration phase, you can readily facilitate compartmentalization, where these lipids will encapsulate the growing polymers, informational molecules. And more recently, some studies have shown that if you start off with double-stranded RNA molecule, RNA is thought of as a molecule being extremely touchy to hydrolysis, right? It breaks down very readily. But if it is already in a double-stranded state, it is much more resistant to hydrolysis, which is what was shown in this study. So given all of this, one of the things we do in our lab is to try and understand how the molecules of an RNA world came about. We try and understand various parameters on oligomerization, as is shown here: temperature, pH, lipids, monomers, drying time. We try to understand all of these factors and how they impinge on oligomerization. We try to standardize reactions for maximizing their yields. And we also have gone on to, instead of working with just one single monomer at a time, we have made our lives really horrendous by adding all of them at the same time, because then now the complexity of the reaction, the products that you get from the reaction is crazy, but we have been able to do that too. This is the setup and reaction. It just looks extremely fooey, because this is supposed to simulate your volcanic geothermal pool, and we can do it just with a heating block. And here are your monomers. This is a lipid, and this is a representation of your monomer, and we facilitate these wet-dry cycles to do these reactions in the lab. We characterize it using few different methodologies. HPLC analysis allows us to look at the nature of the oligomers that are formed. Mass analysis allows us to systematically characterize them and show that it is indeed a dimer or a trimer or a tetramer. And then it's extremely important to also remember that your lipid should be able to retain its capacity to form membranes over multiple of these wet-dry cycles. And if you work with phospholipids, which are complex, I understand, but at least as proof of principle, it was very heartening to see that even over multiple wet-dry cycles, you can have these phospholipids be very stable and retain their capacity to form vesicles. And we varied reaction parameters. I'm not sure if I should go into details, but basically I think this addresses one of the questions that was raised. Only at very acidic conditions can you have this oligomerization happening. Yeah, these multiple peaks depicts higher oligomers. You change the pH from 2 to 3. Suddenly the oligomerization is not favored, and it just becomes worse as you get closer to neutral. So yes, the physicochemical parameters of the space have a very important bearing on the reaction. Nature of acid, some acids do much better. Sulfuric acid, not surprisingly, is the best because it's a very good condensing agent. Temperature has an important role to play. Water, you can knock off water between two molecules only around 100 degrees C, right? So not surprisingly, you have these higher oligomers, right? Yellow guy, black guy, red, and these blue bars going up at higher temperatures. Lower temperatures, you're predominantly still being dominated by the monomer. I'm not talking about this breakdown, but I'm happy to talk about it subsequently. Drying time. Depending upon the drying time, your distribution varies, but 1 hour is enough to strike a balance between the breakdown peak and the oligomer peak. So we kind of standardize all of these conditions, and we work with all of them independently, and you can see that they can oligomerize. But if you notice AMP and GMP, which are your purine nucleotides, for those chemists in the house, this B is the breakdown peak. You see, there's a huge breakdown peak with increasing number of cycles, which doesn't happen with the pyrimidines. Yeah, we can talk about that later. You mix all of them, you still see all of this happening. So what we finally systematically showed is that canonical nucleotides, your modern AUGC containing nucleotides, might not have been the monomers that might have readily formed these polymers of an RNA world, further strengthening this pre-RNA world hypothesis. So what might these alternate heterocycles look like? Yeah, and this is not a question we are asking for the first time. People have been working on this since the 70s. Leslie Orgel, one of the fathers of the field, right? Think of any experiment you want to do in prebiotic chemistry, he might have tried his hand at it. And he basically showed that forming an AMP and ribose condensed nucleoside is a very, very poor yielding reaction. The yield of the product is just 2%. Yeah. Since then, there have been a bunch of us in the field who've been thinking about these alternate heterocycles. And here is an image of about several of them. You can draw them with using pen and paper, right? What are you looking for? You're looking for informational molecules like A, U, and GC, which have these exocyclic atoms that can potentially involve in a hydrogen bonding. So you can draw multiple kinds of purines, you can draw multiple kinds of pyrimidines, at least theoretically. Now you systematically think about what might be plausible. Can they be synthesized under can they be synthesized under prebiotic conditions in the laboratory? Are they found in meteorites? Can they form nucleosides? So now you can systematically rule out many of them and zero in on a bunch of them that are now becoming very interesting candidates. We took some of these candidates and we actually tried to understand if they can form nucleotides in the lab using your ribose and phosphate. And here is to show that we are not the only ones who have tried it. People in the field have tried. This one is a cousin of your uracil, right? It's 6-amino uracil. And here is the formation of the nucleoside with 6-amino uracil. Here is the ribose sugar. They showed that you can form the N nucleoside, you can form the C nucleoside, and you can also form a nucleoside where the glycosidic linkage is on the exocyclic amine group. So we tried to work with a bunch of these, and without getting into too many of the details, all of them worked, but it became very complicated very quick. I'm going to zero in on just this barbituric acid right here to tell you what we did. What is also interesting, quickly to point out, the monomers that I'm showing you here, which look exactly like your AU, G, and C, in this case, U and C. Your U and C, if you just put them in water, they will not organize into higher order structures. But these entities can readily organize into higher order structures. These rosette-like structures can form. So this allows for us to think about alternate ways of how the informational molecule might have formed. So one of the alternate ways to think about this is, like I pointed out, you need these acidic conditions to form the backbone. The same conditions which form the backbone chops off the informational molecule. So then the question is, where am I forming an informational molecule, right? So we then try to understand, can we form just backbone oligomers under early earth condition? And why is this interesting? It is interesting if you sort of think about this hypothetical evolutionary lineage of nucleic acids, right? Your modern biochemistry has this, but it didn't have to be just the same. There might have been something very early on, like a proto-RNA, which has an ionizable linker, IL, which is phosphate in your modern nucleic acid, trifunctional connector, which is the ribose in your modern nucleic acid, and recognition unit, which are A, U, G, and C, and T in your modern nucleic acids. But these could have been made of any number of different things. So one of the important stopgaps in this evolutionary lineage would have been formation of these phosphodiester backbones. And this is something that we wanted to try and see if we can do it in the lab. And we went on to show that you can actually very readily form these phosphodiester backbones. And we also went on to try and show that you can condense the non-canonical bases onto these disposition of the sugar to form informational polymers. So the key findings from this work, the condition which allows for oligomerization of the backbone chops off the informational molecule. So this forces us to think about whether the genetic alphabet as we see today, was it a late period selection pressure? And early on, were there other molecules that might have been present on the earlier that did very similar functions? So canonical nucleotides, not the most fit ones under prebiotic conditions. Then that is not all bad, because then you can have non-canonical nucleotides that do this readily. And we did this in the lab. We went on to show that barbituric acid can condense onto this ribose phosphate backbone to form a non-canonical nucleotide. And these things are cool because just by themselves, right? This is barbituric monophosphate and this is melamine monophosphate. They can form these higher ordered structures, which then can provide some sort of a scaffold onto which now you can constrain the backbone, and potentially that polymer fusion-like model could have also resulted in informational molecules, not just the current classical way that we think about it. So other nucleobase analogs form nucleotides and also oligomerize under the harsh conditions of the early Earth, could have been possible ancestors of modern bases. And we went on to show that you can form these phosphodiester backbones. And this is the classic model, right? Where you have the starting nucleobase, the information entity. It reacts with the sugar to form the nucleoside. And then it probably got phosphorylated to form the nucleotide. And then they oligomerize to form the polymers. And it's becoming increasingly clear that this model of polymerization is really not going to lead to molecules of an RNA world. So what do? We think about non-canonical bases that can readily form these kinds of monomers, and then we can check, do they polymerize? The answer is yes, they do polymerize. When you have the canonical kind of base forming a nucleotide and oligomerize it, you're going to start forming these abasic oligomers where the information is not intact. So then this leads to polymer fusion model that I was alluding to, where you have the monomers, you can readily form the backbones, and onto these backbones, you could think of how non-canonical bases would have been constrained or condensed to form informational polymers of a pre-RNA world. And we have demonstrated this too, yeah? How much time do I have, Vamsi?
Sorry. Yeah, maybe like 5 to 10 minutes more.
Okay, then I'll just skim through some of the other things which we are interested in the lab. And it's as follows. Yeah, so our work basically is underscoring the fact that your conventional polymerization models might not necessarily be the only way you would have had informational polymers form on the early Earth. Now, once they do form, and more recently, we've shown that if you change the starting material, you go from a 5' nucleoside monophosphate to what are called cyclic monophosphates for the chemists in the audience. They result in the formation of intact polymers. And this is something that we very systematically showed. And here are oligomers formed from AMP monomers, and here are oligomers formed from CMP monomers. And you can form oligomers up to 4 nucleotides long very readily. And that was very cool. So why is this interesting? Because once you get to the space from the prebiotic pool of nucleotides and you non-enzymatically can oligomerize and form these varying length oligomers, then the immediate next thing to think about is how might the information in these molecules be transferred? And this is of super important consequence, especially if you have formed functional molecules in the space. And this is something that is eventually important to understand because it has implications to think about how did the first ribozymes systematically emerge. Or you could have had it emerge even here, but maybe it might have been a much simpler catalytic molecule. You need a reasonably long size molecule to evolve functional possibilities or catalytic possibilities, because it should be able to form this nice pocket with which it can bind the substrate, and very short oligomers can't readily do that. So one of the very important thing we're very interested in is to understand the role of prebiotic complexity in all of these processes that we're talking about, right? So this is a paradigm that we have worked with. This is called templated replication. So let's say you formed this template and the primer readily from the kind of reactions that I was talking to you about. You could now think about faster reactions that can be facilitated once you have this primer template complex, which then can quickly allow for the zipping up of the remaining information that is on the template by using the remaining information on the template to form a new strand. So in this paradigm, we try to understand the role of prebiotic complexity on non-enzymatic replication of RNA. And what we found is that when you have co-solutes in the reaction, typically what the field did, they tried to just understand the process. Template, primer, nucleotide, facilitate the chemistry that is required and understand the process. Very important to understand the fundamentals of the way it panned out, but it is not realistic. Prebiotic soup was anything but a homogeneous mix of molecules, right? It was a heterogeneous hodgepodge of molecules. So we systematically try to complexify the reaction to understand what might these processes, how might these processes have panned out in those scenarios. And what we are seeing is that there is much higher mutation rate in these scenarios. And is that all bad? The answer is no, because much higher mutation rate on the early Earth means a quicker possibility of sampling all possible sequences in shorter number of cycles. So it's all not bad. And is this something that I'm just faffing around? But no, we actually did studies, theoretical simulation studies with colleagues. One is in IISER Kolkata and the other one is in University of Paris, and went on to show that you can have these more complex secondary structure containing RNA emerge much more readily when you account for these higher mutation rates, which is sort of fantastic, yeah? And what else are we interested in understanding? The fundamental biophysics, the underpinnings of the process. We use biophysics to understand some of these processes which are playing out in these co-solute driven reactions. And here we have been collaborating extensively with JEET, where we use NMR as a tool to understand these processes. So this is just a figure to show that this is not surprising at all, but nobody systematically looked at it very seriously. But this is at least from a prebiotic chemistry perspective. So if you have co-solutes like PEG in the scenario, your diffusion of the monomer is affected because it now starts, particularly your purines can form these pseudo oligomers, and the more the crowding there is, the more it seems to be affected. And this we showed using some pool NMR stuff with at least some proof of principle results have come out with our studies in conjunction with GTube. And in conjunction with Raghu at RRI, we've gone on to show that the RNA lipid interactions that are facilitated in these scenarios are completely non-trivial. Depending upon the nature of the nucleotide in question or the monomer in question, it could be anything, it doesn't have to be a nucleotide, the kind of events it facilitates is very different. So in one case, the stop panel is basically UMP, yeah, uridine monophosphate by itself when it is present in the when it's in 1:1 ratio with DMPC. You can see that it can form these unilamellar vesicular structures. But now if you have a disodiated version of the UMP with the DMPC phospholipid in 1:1 ratio, it now results in large multilamellar vesicles. So all of these things have fundamental implications for the kind of concentration mechanisms that I was talking to you about. And these are things which we are just beginning to scratch the surface of. And in more like super hot off the press results, we also looked at oligomerization of amino acids to result in peptides under early earth condition. And we showed that when you do these reactions in the presence of other co-solutes like lipids, if you just take the amino acids and do wet dry cycles under certain conditions, you can form peptides. But now you're thinking about processes that eventually led to the formation of the protocell. So you have to account for these other co-solutes that are fundamentally crucial to the formation of the protocell. So you put them in this reaction mix, and what happens? You do form the peptide, but there is a competing reaction that kicks in that forms what are called these N-acyl amino acids. Yeah, this is essentially just like your fatty acid, but the head group is that of a amino acid. Yeah, so you can have peptide synthesis. You can also have the formation of these N-acyl amino acids, and this is just something that we published literally a couple of days ago. And coming to the membrane part, we are very interested in understanding the evolution of model protocell membranes. And in this context, we are looking at what are the different entities that might have made the landscape of the lipid molecules or the amphiphilic landscape that might have been available for formation of these protocells. Do some provide a selective advantage over the others? Are homomembranes better than heteromembranes or vice versa? There's no reason to believe that homomeric membranes would be better. And we actually went on to systematically show that compositional heterogeneity in this protocell membrane actually has a selective advantage. So I think I will stop with this, and I'm happy to take questions that then can delve into some of the more systematic systematic work that we did.
Okay, yeah. Thank you so much, Professor Rajamani. I think we'll take a few questions here. First from the chat box, I guess. So I see one question by Michelle. It's a long question. Michelle, can you unmute yourself and ask the question? Okay. Michelle, are you there? Maybe she's missing. Okay, I'll go to the next question by Saurav, which is at what stage and how does consciousness develop in these molecules? And why does the definition of life not require the entity to be intelligent or conscious?
See, for the fundamental transition to occur, I don't think one needs to invoke cognition. However, in order to be able to understand this crazy sort of how do I say, there's this orders of magnitude difference when you think about cognition between your unicellular entities and humans, right? With this whole spectrum in between. When you now think about that kind of a change in complexity, which is much closer to the emergence of more complicated entities, literally in the sense of complex in the form of their shape and their size, so to speak, not shape, their size. One probably could think about invoking cognition. I do not know where in this billions of years history, early history of life on Earth, can you fit in cognition? It is something that I have peripherally flirted with that idea, but I really am completely out of depth here to be talking about that. But at least for understanding the transition from chemistry to biology, one doesn't have to invoke the origin of cognition at all. Yeah, because you can actually have just physicochemical properties drive interaction of these vesicular entities made up of synthetic lipids in the lab. Synthetic lipids, vesicles made from synthetic lipids in the lab have shown processes which are lifelike, right? They can grow, they can divide. There is even a primitive version of evolution that has been very elegantly demonstrated in these vesicular populations. Do they have to be intelligent to be able to show these lifelike properties? I think the answer is no. They absolutely do not have to have a intelligent cognitive mind to be able to do that. That is like a very, very complex thing that might have probably kicked in much later on, very close to the emergence of very large organisms, right? So yeah, out of depth, I do not know where to place it on the scale, but remotely nowhere in the time scale that I was talking to you about. It might be very, very close to, I don't know, little older than dinosaurs maybe, but even there, I really do not know.
Okay, thank you. And Professor Shubha Thole has a question. By what test would you determine if a bacterium has consciousness?
No, it's not a question. It's a response to the previous chat message.
Oh, okay.
Okay, so get it. Sorry. Yeah. And Professor Alakre has a question. Are these non-canonical protonucleotides found on meteorites?
That's a very interesting question, right? So some of them have been found on meteorites. Not all of the ones that I, actually, let me go back. You'll notice that I didn't go through more of the slides that I wanted to share with you, but that's okay. Where are you, buddy? Sorry, one sec. Yeah, there we go. Yeah, so you see the purple square here? That means you can either find them in meteorites or they have been demonstrated by syntheses under prebiotic conditions. So you have this whole bunch of them, including barbituric acid, which has been found on meteorites. So yes, some of them are found on meteorites. Here is one with a purple box, but a cross. It essentially means you can find this on a meteorite, but it doesn't show any hydrogen bonding. So then that rules it out as a probable candidate. So, but yes, the answer is some of them have been shown on meteorites.
Okay, thank you. And then I think Sriharsh has raised his hand. So Sriharsh, you could ask a question.
Sorry, can you hear me?
Yeah, I can hear you.
Okay. I had two questions. One was, what is the evolutionary pressure to move from these non-standard nucleotides to the ones we have now? Is there, what sort of advantage do these provide, and what sort of changing conditions led to those? The other question I had was about transient events from the parent star. So M dwarfs, for example, have a lot of UV flares, or the conditions on the planet can change substantially for short periods of time. Do those have any particular effect on this kind of chemistry and biology?
The second one I will speculate about, right? Because I'm really not an expert in that area.
Absolutely. Yeah, I would love to hear your thoughts.
So one of the important things to remember is a lot of these molecules which are pertinent to extant biochemistry are UV sensitive. You do have actually radiation driven damage happening in our cells depending upon the kind of exposures that you have, right? And there is this correction machinery which kicks in, which tries to make sure that it repairs it and makes sure that it's weeded out of the pool. So yes, if all of these events were happening on a planet where some of these the turnaround of some of these events are very quick and the kind of conditions were really harsh, it will surely have a bearing on some of these processes. There's no questions about that, and it is going to heavily impinge on the stability of the molecules that can be formed, some of the molecules that can be formed under these conditions. Going to your earlier question, which is something that we think about a lot, what are the evolutionary constraints, right? I will answer your question first in the following manner. So this particular thing, let me go back to the key findings. Yeah, so here, this is barbituric acid right here, yeah? And we could show that it can form a non-canonical nucleotide. And when it forms this non-canonical nucleotide, you're forming a mix of products. You're forming a alpha CBMP, you're forming a beta CBMP, and this is just the glycosidic linkage, which is a C-glycosidic linkage. You are also forming N-glycosides in the mixture. And if you look at your modern nucleotides, this glycosidic linkage is an N-glycosidic linkage, which is much more labile if you veer away from your ambient pH in the cells, your physiological pH. So the thinking is that those nucleotides or those non-canonical nucleotides, precursor bases or ancestor bases, barbituric acid is actually even found now. So these kinds of molecules that can form more sturdy glycosidic linkages might have had an advantage during the very early times when the conditions were harsher. Yeah. So with time, the conditions have gotten more ambient, so to speak. So this decrease in the harshness of the environment would have been a very important selective pressure that eventually allowed for the survivors that we see today. But whether, see, even now, your AUGCT is what you predominantly talk about. Even in modern cells, you have this whole bunch of modified RNAs that exist. The modifications, though, might be a result of an enzyme's action. But having said that, it is clearly sort of indicating to a time at an earlier point in the origin of life, or in the evolution of life rather, early life, where we probably had many more different kinds of these molecules. But it is these modern extant bases that you see today, not just AUGCT, but these other slightly modified versions that eventually turned out to be the winners. So some of the selection pressures could have been pH. Other kinds of selection pressures could have also been divalent cations. Divalent cations is a very important selection pressure to factor in. There is this very sort of tight regime in which it is fantastic. It is required for, I'm going to use protocell formation as an example, right? For protocells to form, you require a certain amount of these monovalent and divalent cations, because if you just look at fatty acid protocells, for fatty acids to form these vesicular structures, these vesicles, it happens at the pKa of the head group of the acid. And at that pKa, you have both protonated and deprotonated entities. And for some of these entities to kind of stay close to each other and form the stable structure, you need some amount of counter ion effect to be able to stabilize them. But now increase the concentration from these tens of millimolar to higher amount of mono or divalent cations, and you're going to have them fall apart. Your ribozyme or any of your modern enzymes require monodivalent cations in a certain concentration. I'll take ribozymes as an example, yeah, made up of RNA monomers. So you need a couple of tens of millimolar of magnesium, for example, for the hammerhead ribozyme, to facilitate its catalytic activity, to self-cleave itself. But now you change the concentration of this of magnesium to much higher amounts, your RNA will hydrolyze completely. So some of the, there were billions of years for these selection pressures to play out, right? What exactly played out at what time is very hard to guess, but it is reasonably logical to envisage that there might have been a lot of these systematic selection pressures, depending upon the niche where these reactions are happening, that would have allowed for shaping some of these events to a point that we see where modern biology is at, right? I mean, especially 2 billion years ago when you had, or 3.5 billion years ago when you had the actual transition from chemistry to biology happen. Even then you did not have the most conducive atmosphere, but it was much closer to modern atmosphere than it was to early Earth. So even there one can start sort of narrowing down or constraining the parameters for these selection pressures to some reasonable extent to then think about what they might have been that might have allowed for these transitions to happen. Am I making sense?
Yeah, that was a great answer. Thank you.
Okay, thanks, Harsha.
Okay, so Arya has a question. Arya, you could unmute and ask. Arya Nachiketa. Okay, maybe
I'm sorry I didn't get to talk about the protocell work much. I think that is the stuff that JD was most excited about, but I'm happy to talk about it with those of you who have any specific questions about that. But I would like to just say that what we have shown is the following, and let me just go to sort of the main crux. We've been able to show that if you have a compositionally heterogeneous system, you can actually increase the pH regime over which they form these stable membranes. Now, this goes back to an important question that Vamsi asked very early on. And the ramification is that all of the prebiotically relevant processes don't pan out at the same pH. Some things pan out at a lower pH, some things pan out at a higher pH. And so it's extremely important to try and understand whether you can form robust membrane systems that can tolerate a wide range of pHs if you really want to understand the emergence of protocells as a process, as a phenomenon. So for that reason, we are very interested in understanding the compositional diversity or the landscape of all the amphiphiles that might have been available for these early protocellular membrane formation. And given that a few diverse kinds of them would have been readily plausible, we are very eager to understand what combinations of these molecules are much more robust. For example, here is oleic acid. If you take just an oleic acid vesicle, just an oleic acid based monomeric pool, and if you're above the critical vesicular concentration or the concentration above which they can form these higher ordered structures called vesicles, they can form vesicles only within this very tight regime of pH. But then spike it with other derivatives of oleic acid where the fatty acid head group is now replaced with an alcohol or a glycerol monoester. You can form these binary systems, or you can actually mix all three of them together to form a tertiary system. You now change the regime in which you can form these thermostable protocells. And this is extremely important for us in the field to kind of understand, mainly because it has fundamental implications for not just the emergence of protocells, even for the evolution of these outer compartment.
okay. Thank you. And maybe a couple more questions. Sudipta has one question. Sudipta?
Yeah, hi Sudha. That
Hi.
was a fantastic talk, and of course, one of the most interesting questions that you can ask in science. I had a question that I have put in. So there's a probabilistic event. Okay, so the fact that these things would get together, would get enwrapped by in a vesicle or so. So it is something that will happen occasionally. So has it happened on more than one occasion?
Has it happened on more than one occasion?
Has life evolved more than once on this planet?
So
what does calculations say?
So this is a very tricky question. So is this you're now almost asking a question, is it a probabilistic event or is it a deterministic event, right? So the way we think about some of these transitions allude to the fact that it is a deterministic set of events that would have been, that would have played out in a certain time frame. But once you moved beyond that time frame, see, for example, I will again use this fatty acid, like you pointed out, formation of fatty acids on modern Earth, right? You might be able to form them in certain niches of the modern Earth. You just have to invoke extremely high temperatures and pressures. I can think of niches where you can invoke one or the other, not the two of them simultaneously. So I feel like, yes, they are very readily demonstrable in the lab, but to think about actual analog environments where these kinds of processes can be facilitated super readily is non-trivial, with fatty acids in particular, right? The fatty acid formation. But the story is different for the nucleic acid based polymerization, nucleotide polymerization. I can go to a volcanic geothermal pool where the temperatures are high. They are higher than 75. And in the lab, we are showing that above 75, you can actually form these oligomers. And there is a possibility that I can, in principle, be able to demonstrate the formation of this oligomeric nucleic acid from the starting monomers. However, there is one problem that is there that I haven't even touched upon. And the problem is the fact that we really do not know the dimensions of these pools in these analog environments, Shudipto. So when you pour an X concentration of your reactants into this pool, very quickly they disappear. So there's no way of tracking them. But having said that, given that a lot of these environments are clay rich and clay lined, if they go and sort of get concentrated in reasonable concentrations in the environments, maybe it's possible. But the kind of concentrations we're talking about for facilitating these in pools which are even a few feet wide is huge. So I do not know if these processes are happening on modern analog environments as we speak. But for example, the fatty acid business, very difficult. On early Earth, you had very high temperatures. And if you were you had shallow oceans, so you would have been able to very readily form them in some of these vent-like areas where you could facilitate these Fischer-Tropsch type syntheses, right? High temperatures, high pressures. But now I can't think of, maybe at the core of hydrothermal system, it might still be happening. Do not know. Do not know. Possible, right? If even now you have these acidic and alkaline vent systems, and at the core, the temperatures are very high. They are going to much more closer to the magma phase, right? So the temperatures are very high and the pressure there is very high. Is it happening even now? I do not know. Possible.
Thank you.
Okay. I guess it's time, so we'll end the session at this point. Thank you so much for this exposition. This was very, very interesting to us. And I hope everyone can give a round of applause to Professor Sudha Rajamani. Thank you so much. Yeah.
Thanks a lot. Thank you. Thank you for having me. Like I said, I wish I did this in person.
Yes.
I got bombarded with much more questions in person, many more questions in person, but hey, another time. Okay. Thank you so much.
Thank you so much. I request you to stay back, and also Professor Sandhya Kaushika, I guess she would meet you. Okay.
Thank you all for.
Yeah.
Sorry, I said your name correctly, right? You're Vamsi.
Yeah, Vamsi.
Man, I said it a few times and then I realized, I was like, shit, am I saying the wrong name?
No, it was great. Yeah, it was great. Thank you. I was actually surprised that you remember.
So I always give a second name to most people that I meet. It happens very early on during my interactions. Okay. I'm glad it didn't happen this time. It would have been really embarrassing.
Yeah, this is great. Yeah. Thank you.
Hello, Sudha.
Hello. How are you?
I'm fine.
Oh, wait, let me stop sharing, no? Chalega kya?
Haan, chalega. Totally chalega. Let me turn on video. I sort of missed the first 20 minutes. There are people. Who's the host?
Yeah, it's me, Vamsi.
Oh, okay. Hi, Vamsi. Can you maybe remove the other participants?
Yeah, I'll try to do that. I request others to exit. If not, I will slowly start removing them.
I'm happy to meet you all another time at your convenience.
I have a feeling that some of them might have just stepped away briefly. And
yeah. So I'll just remove them. Making sure I don't remove others.
Please don't remove me.
Yeah, exactly.
Not that I can always rejoin.
You won't have missed too much. You already have a good kind of notion about a lot of the context that we use, in whose backdrop we work, because you do quite a bit of reading in this area. So I don't think you missed much at all.
No. I've now kept the YouTube link. This is my job. As soon as I finish speaking to you, I'm going to rewatch the first 20 minutes. You can't believe I finally went and, I had to do a bunch of stuff. I'll tell you what. I went to get a Wakalatnama signed. Today's last day to post it for the Sci-Hub case.
Oh,
So for Sci-Hub and LibGen, there are a bunch of...
It's interesting.
Okay, thank you, Vamsi. It was very nice moderation. Actually, he asked me to...
Vamsi, can I ask you for a favor?
Yeah, sure.
Be able to send me the questions again, if that's okay?
No, just download the file.
Oh, I can.
In the Zoom window, you
I
have
can
the file, no? You can just download it. So you'll have all the questions.
Oh,
The chat, open the chat, and you will see file, right? And beside file, you will see 3 dots.
I saw that, yeah.
And then you see save chat.
So
No.
I have saved it in case you
if I did a disastrous job, I'll get in touch with you.
Yeah.
Yes. But if you just save the chat, it'll all be there.
All right. Thank you, Sandhya. I have made you the host. I'm not
okay.
sure if anyone else is meeting Professor Rajamani.
I don't think so. So JB told
me
just Sandhya.
Okay.
Okay. Because there was some sort of schedule, and I guess no. I'm happy to chat with her.
Okay. Bye, guys.
Bye. Thank you, Vamsi. Very nice moderation.
Yeah. Bye.
Hello. How are you?
Hi. How are you?
How am I? I'm good. Hanging in there. I'm just kind of reaching a break point. I need to get out of IISER Pune and take a break. So other than that, I'm fine.
Take a break, na? You can go out. You can take a hike or something. I always see these amazing pictures that Nagaraj puts up on YouTube, on Twitter. He always seems to be doing exciting stuff.
Nagaraj does exciting stuff all the time. So with me, I'm actually planning to take off on Monday so that we can go for a little hike and stuff.
Yeah, sounds good. Actually, today I went out to do this Wakalatnama, and then I had to post the Wakalatnama because the lawyer actually said that we have to send everything on the latest by 20th, because it will show up. We have to, submit those things.
Hey, Sandhya, we are still live on YouTube. It's saying live on custom live stream.